Hi everyone, my name is Stefanie and I'm a PhD student at the University of Sydney. I'm also on the committee of the Royal Society of Biology Australasia, and I normally deal with social media and communications. So today I would like to start my presentation by acknowledging the traditional custodians of the land on which I work, which are the Gadigal and Ngarigal people. And I'd like to pay my respects to their elders past, present, and emerging. And now I will go on to talk about carcass ecology in Australia. So some of you may ask yourselves, why study carcasses? So we can study them for forensics, which helps with solving crimes, but that's not our focus. We focus on the ecology of it, where carcasses are considered a resource. And carcasses can affect food web dynamics and disease transmission, which has consequences for biosecurity, public health, and land management. So our lab has been studying Australia's scavenging food web, which is represented in this figure. And this food web includes both native and invasive species. So the invasive species are a conservation issue in Australia. For example, herbivores such as deer and horses can damage soils by trampling, but they also can overgraze and overbrowse vegetation. Introduced predators such as foxes and cats can cause massive damage by predating on native animals, which do not have anti-predator mechanisms against them, so they cannot defend themselves. So for this reason, a lot of carcasses that are found across the Australian landscapes are a result of land management, so culling, which is why we've got a deer in the middle here. Australia's scavenging food web, the scavengers are comprised of aerial scavengers such as wedge-tailed eagles or ravens. We've got terrestrial scavengers such as goannas and dingoes. And then we've got some introduced ones as well, such as pigs and foxes. Invertebrate scavengers are also very important for getting rid of biomes. But a carcass does not just affect the animals that feed on it. It is also a very important part of nutrient cycling. So when we get a carcass, nutrients flow from it into the ground, and this can either kill vegetation or promote its growth. And then the vegetation that grows up can also be eaten by herbivores. So the methods we use to study carcasses and scavenging food webs are similar for the studies we have done, but they may change slightly depending on the questions we ask. So we use camera traps to monitor vertebrate scavengers. We use pitfall traps to assess invertebrate communities. We also collect soils and we can put soil probes out sometimes as well, and vegetation samples. In some of the studies, we also exclude certain types of scavengers just to see what the effect is of not having certain animals eating carcasses. So the first study we're going to have a look at is by Emma Spencer. Emma has spent the last 4 years monitoring carcasses in different biomes for a total of 272 carcasses monitored over 12 study periods, which is pretty impressive. So what Emma has done is she put carcasses out in the Simpson Desert, which is a desert biome, of course, in the Blue Mountains, which is a temperate forest biome, and in Kosciuszko National Park, which is an alpine environment. And this photo here shows what the setup is like. So you've got the carcass and you've got the camera trap pointing at it. Emma's study specifically looks at scavenging by dingoes. And this graph shows feeding events in the black bar over here. And in the white, it shows carcass visitation without feeding. Emma has looked at this behavior in closed canopy habitats and open canopy habitats. And she's done this in cool and warm seasons as well. And she's done this across all of the biomes. So we can see that dingoes visit carcasses quite consistently in temperate forests, but they show a slight preference for feeding in cooler temperatures. This is different from alpine areas where dingoes show a preference for feeding in warm temperatures, and they show a preference for closed canopy habitats. In the desert, instead, they don't show up in warm periods. However, they show up in cool periods, and they show a slight preference for feeding in open canopy habitats. And this might just reflect the differences in resource availability among all these biomes. The next few studies we're going to have a look at are all carried out in Kosciuszko National Park in the Snowy Mountains. This is a fragile and unique ecosystem in Australia. And carcass research here is important because we want to know the impacts that carcasses have, as culling is a prevalent land management technique to control feral pest numbers. So the studies we're going to have a look at now, they have not been published yet, but we'll have a look at some preliminary results. So here we are having a look at what Chris Fast and James van der Steen have been doing for their masters, and their studies involved placing carcasses along altitudinal transects. This plot that we're seeing over here is a survival analysis plot. So survival analysis is used a lot in population studies, where you look at factors that affect the survivorship of different cohorts of animals, but it's also used a lot in medical research for clinical trials, for example. So we can consider a carcass as a finite resource, which starts from 100% and finishes at 0%. And the 0% can be considered as the death point for the carcass in survival analysis terms. So in this graph, the 4 different lines represent seasons. And the steeper the line, the faster the carcass goes. The flatter the line, the more the carcass persists in the environment. So we can see a clear difference in summer and spring. The carcasses go a lot faster, whereas in autumn and winter, they stay a lot longer. The next part of the study was looking at some exclusion studies. So vertebrate exclusion was achieved by using some mesh cages, and invertebrate suppression was achieved by using fly screen and mesh. This graph shows that invertebrate suppression resulted in a lot longer carcass persistence, whereas the difference between vertebrate exclusion and no cages was not that much. So this is due to the highly variable nature of the data. The open carcasses that didn't have any cages on, they were not all visited by vertebrate scavengers. So that's what caused the variance. However, when they were present, the carcasses went a lot faster. The other thing that this graph shows is that invertebrates can actually be very important in getting rid of biomass as well. The other thing that we've all noticed when we were studying carcasses in alpine environments was the presence of European wasps, which are invasive in Australia. And we've all seen them decapitate blowflies, as you can see in this little video. They just grab them and chop their head off. And Emma Spencer, in one of her studies, has shown that when the numbers of wasps in pitfall traps are high, then the numbers of blowflies are actually quite low in the pitfall traps, but you get a higher proportion of them that have been mutilated. Emma has also shown that larger vertebrates can also be affected by the presence of European wasps. As a matter of fact, you can see dingoes being harassed and their feeding behavior can be altered by the presence of wasps, while invasive scavengers such as pigs are not really that bothered by them. So this shows how European wasps might actually be an important pest to have a look at, and we need to study it more to know what impacts it has on scavenging food webs. So next, we're going on to mass mortality events, which is actually the topic of my own PhD. So mass mortality events have been increasing in the last 40 years. Some of them may be natural events, but the frequency at which they are increasing is not natural. So part of this has been linked to climate change because, of course, it makes extreme weather events more frequent. And part of it can also be attributed to anthropogenic activities. For example, oil spills or habitat degradation can cause mass mortality events. So the problem with these events is that they produce a large amount of carcasses in a small period of time. And this begs the question of how they are managed. So procedures for dealing with these events are quite clear when it comes to farmland and when it comes to publicly accessible land, but they're not quite as clear when we get a mass mortality event with wild animals or maybe in fairly remote areas. So the aim of my PhD is to determine what the environmental effects of different loads of carcasses are. And I'm doing this by looking at plots with 10 animals and comparing them with plots with single animals, and then I've got no carcass controls for this. So I also use exclusion cages to see how these effects change when you exclude different types of scavengers and how heavily this impacts the environment as well. So far, I have put out 88 carcasses, which is half of the target I have. And all of these have been out for a while, so they have reached the decomposition stage of dry decay or skeletonization, which means there is very little tissue left and bones are showing now. I've got lots of data to process. So I've got camera trap data, I've got invertebrate data, I've got swabs that I've collected of the carcasses, which have been sent for RNA sequencing. I've got VOC tubes, volatile organic compounds, and soil probes, which are ion exchange membranes. And then I've collected some vegetation data as well. So I'm not looking at all this data myself. Actually, some other labs from University of Sydney, but also Federation University are looking at some of this data. And so hopefully we're going to have quite a detailed picture of what happens when you get these mass mortality events by the end of it. So some observations, these are some pictures from camera traps that I had a look at. So we've got the usual suspects in terms of scavengers. We've got our aerial scavengers, such as the ravens. We've got our terrestrial native, such as the dingo. And then we've got our invasives, such as these fox kits, just fighting in front of the camera. We also have herbivores that come in and eat the grass after the carcasses are decomposed. Other lesser known scavengers we've got here are, for example, brush-tailed possums, which are generally considered as herbivores in Australia, although there is evidence from New Zealand that suggests otherwise. But because we've had so many carcasses out, we actually can show that they actually spend a lot of time around carcasses. And we're working on a paper to actually quantify this. So the road ahead for my project is that I still have 88 more carcasses to put out and a lot of data processing and analysis to do. And then I will be able to report on the results to the wider scientific community. So I'll be spending a lot of time in front of a screen once my fieldwork is done. So some of you may ask yourselves, how do you apply this knowledge? So in terms of mass mortality events, we think that my study will be useful for determining whether we need coordinated response teams to go out and collect data or deal with disposal procedures. This already happens to a certain degree, but it's impeded by funding and logistics because you need to get there quite quick, especially if you want to determine the cause of why it happened. The other lot of implications regard vertebrate pest management. So here the goal is to minimize carcass persistence, but it is also to minimize utilization by invasive species. So we think this can be achieved by adjusting the timing of control, and the timing being when the carcasses go faster, that would be the most appropriate time to cull. And We also think there needs to be more targeted fox and pig control, but where possible, we should reduce dingo baiting because they are Australia's top scavenger and they are very good at getting rid of biomass pretty quickly. So hopefully with this short talk about carcass ecology, I've shown you how carcass ecology has many implications. And our studies have shown how variation in decomposition happens among biomes, but also among seasons. And so you need to study them across different locations and different times. And hopefully I've also shown you how knowledge about this can be useful for land management decisions. So this brings me to the end of my presentation. So if anybody has any questions, feel free to ask.